Good morning. I'm Lisa Wiener-Intrator, and it is my pleasure to welcome you here to Deep Tech Health plus Neurotech Boston. It is the first conference that we've done in 31 months. 31 months ago we were at Stanford, and 34 months ago we were at Harvard, and I am really thrilled to be here, and thank you for being here today. A lot has changed in the world. And it's well known that the 10 years surrounding World War II gave us dramatic advances in artificial intelligence and communication and computers. And I think that in a sort of similar way, the positive outcome of the tragedy of our time, this global pandemic, will be dramatic advances in medicine. I think that it's obvious that mRNA has already saved many lives in the COVID pandemic, but mRNA is also now being used for cancer and other diseases. And just this week, we might have had a therapeutic breakthrough in Alzheimer's disease just a few months after the amyloid hypothesis debate. Today, we will have a series of sessions about sensors, AI, genomics, and surgical innovations. We'll talk about better clinical trials and federal and VC funding. As always, we'll have several sessions on the brain, from anesthesia to early detection of cognitive decline to stroke, brain injury and cancer detection and treatment through a wearable, and perhaps a better way of doing brain-computer interface. There have been some speaker changes. We have two additional speakers. David McMullen, who's outside, he was just appointed 3 weeks ago as the head of neurological and physical medical devices at the FDA, is going to speak about the regulatory process. And Ellen Roche is going to speak this afternoon. She's actually not on the printed schedules on your table. She's a professor from MIT doing amazing work with cardiac care centers and soft robotics and the like. She's going to speak at 3:55. Two of the speakers are not able, well, actually three. Bob Langer and Bakul Patel cannot be with us. Bakul recorded a short presentation about Google AI and healthcare, and Bob pre-recorded an interview with me that's really fascinating, so I'm happy that you're going to be able to see it. Between those two videos is going to be Ellen Roche's presentation. And now it's my great pleasure to introduce Giovanni Traverso. Gio is a physician scientist, an MIT professor, and a prolific inventor who Bob Langer spoke about in our interview, actually. And he also made it possible for us to have this conference at MIT, and for that I'm grateful. And I'd also like to welcome the several members of his lab that are in the audience. Welcome, Traverso Lab. So thank you all for being here. I hope that you enjoy the day. I hope that you will find it as fascinating as I find it. And I hope that you'll be back with us again and again. Oops.
So thank you, Lisa, and thanks everybody for joining us this morning and for this incredible day. Let me dive right into the GI tract. And just before I dive in and share with you some advances and work we've been engaged with over the past decade, just want to share my disclosures. As part of our work, we are deeply committed on getting the technologies that we develop in the lab to help patients, and we do that by collaborating with many different companies as well as participating in forming new companies to help with that process. And these are my current disclosures. The first vignette, or really journey, and I would say journey because it continues to this day, is one that started about a decade ago, really through some incredible interactions with the Gates Foundation, and has really involved many key individuals over the years. And here I showed just a few of them. It really is an incredible sort of community that has supported this effort. And really what we're trying to do is change how we can help people receive medication. And the reason that we're trying to do this is, for the very simple sort of recognition that in general drugs don't work if people don't take them. And despite this obvious statement, the overwhelming majority, and this is sort of the statistics here for example, in Boston, in the developed world, is that 50% or more of patients actually fail to take medication as prescribed. This has been written about by many, including us, but this is a report from the WHO highlighting the magnitude of this challenge. And so in the developing world, the non-adherence or non-compliance challenge is estimated to actually impact about 70% of patients. And it's something that's recognized to actually continue to increase as we start to develop better therapies for a range of conditions like HIV and tuberculosis. And probably one of the most compelling quotes I've come across over my time working in this area is the one at the bottom, and that is that improvements and advances in our ability to enhance adherence really stand to have a greater impact than the introduction of new medications, which to me is just simply astounding. Now, as engineers, we started to think about how we might be able to help patients with receiving their therapy. And we really honed in on a very simple and basic observation, and it's one that at least I've experienced in my lifetime, where, for example, if I'm prescribed a medication that I can take 3 times a day versus a similar medication that I can take once a day, in general, I will go for the ones that I can take daily for the simple reason that it is easier for me to remember to stick with the medication schedule. And when one asks the question, is this something that the general population also observes and really demonstrates, what we see is that the more infrequent the dosing schedule, the more likely individuals will continue to stick essentially with their medication regimen. Now, this may seem obvious sort of on the within sort of the 24-hour period, but actually has been extended to sort of longer, more infrequent intervals. And this is a trial out of Japan highlighting that observation for a very specific type of drugs. These are drugs that are used for osteoporosis. They're bisphosphonates. And what you can see here, what's represented on the y-axis is persistence rate in this particular trial that was evaluated as the likelihood of a patient going back to the pharmacy to continue to fill their prescription. And what was observed in this trial was that when the medications were provided at a more infrequent interval, those individuals would continue to fill those prescriptions as compared to the more frequent dosing. Now, similar data has come out of Europe as well as the US supporting this. So with this body of data, we recognize that perhaps it might be challenging to change the individual, but could we change the pill? Could we transform the capsule? And so that's what we set out to do. And the vision when we started, I think was fairly well captured in this graphic from The Economist talking about our work, which I really feel highlights some of the key aspects. One, it's an orally delivered system that we are aiming for. It is one that is providing metered dosing, and hopefully in a smaller form factor than what you see here. And now, in trying to do so, what we recognize, and this is something that we all experience, that as we ingest material, generally food or medications, those materials will transit through our bodies on the order of a day or just over. And so the challenge becomes one of how do we overcome the natural physiology of gastrointestinal transit. And so what we were trying to do is really we started with a standard capsule, which typically one ingests, and then its contents are generally absorbed in the small intestine. We can start to delay the transit time by, for example, using mucoadhesives and engaging that mucosa more avidly to slow down that interaction. That generally will give us a few hours, essentially, of extended residency. But really what we wanted to do was have systems that remained in the body not for just a few more hours, but rather for many days, weeks, and potentially even years after a single ingestion event. And so with this in mind, we started to break down the challenge that we had. And the first challenge that we recognized was that we have a ship-in-the-bottle problem, meaning if we want something to stay in the GI tract, first we have to transit or traverse the esophagus, so the neck of that bottle. So we need to fit this object inside of a capsule. So after that, whatever's encapsulated needs to deploy. What you're seeing here is one of the very early experiments of some of our prototypes deploying in the stomach of a pig and assuming the shape that it was designed to assume. Then whatever is deployed needs to stay there, and it needs to stay there for a prolonged period while protecting the drug. And you need to recognize that this is an extreme environment with pH variation between 1 to 5 that is exposed to the variation of the human diet. And then all the while sitting in this extreme environment, we want these systems to release a drug in a controlled fashion. And then eventually, whatever is retained, we want to essentially excrete. And we want to have some control over that transit or passage. And so when we started, we often think about the worst case scenarios. And so in our case here, we wondered, well, what if these systems passed through the stomach and went into the small intestine? And so as part of this, some of our early efforts looked at developing new materials that could provide that safety or a safety trigger such that if there was an inadvertent passage, that object, that macro structure, the star or some other kind of object, would break up so that it could more easily pass. And so we developed early on an enteric elastomer. And that term enteric refers to the very specific property of a material that is stable in the more acidic environment. And what you see there on the left is essentially mass on the y-axis and then time on the x-axis. And when that material is in SGF, or simulated gastric fluid, which is acidic, it remains stable. But when present in the near neutral environment of simulated intestinal fluid, or SIF, it starts to break down. And it is elastomeric because it can be stretched. And we needed it to have these properties to impart the safety aspect, but also to impart the flexibility and compressibility to accommodate the encapsulation of the system. This allows us to actually start iterating in a significant or a meaningful model, in this case the pig, which resembles in dimensions the adult gastrointestinal anatomy. What you see here is one of those early prototypes in a circular form that allowed us to take that material, that enteric elastomer, interface it with another material called polycaprolactone, or PCL, which is an FDA-approved material that can be loaded with drug. And then we showed that we could place it, for example, in simulated gastric fluid, and that those shapes would resume the shape that they were designed to assume, and if placed in that near neutral environment, that macrostructure could break down. Now, we started to test some of these early concepts in pigs, and what you're seeing here are those early serial x-rays of two different concepts. Early on, we had a star, we also had a hexagon, and we looked at the deployment from a capsule. Just to give you an up-close and personal view, this is a really early video of one of those early concepts. And what you're seeing is that star overlying the exit of the stomach, or the pylorus. What I want to highlight is that the mucosa is pink, it's healthy, there's no bleeding, there are no abrasions, there's also no food. And so this is an animal that underwent an overnight fast. And it's really important to appreciate that these objects are not causing what we refer clinically as a gastric outlet obstruction. So simply, they're not holding up any of the food. And this was really reassuring. And this system had been in the stomach of this pig for 6 weeks. Now, in addition to this experiment that I'm showing you, there had been another 300 that had been performed. Now, this was all really reassuring and gave us a handle on how we might contemplate delivering something for a prolonged period, but what to deliver. And this is work that was funded by the Gates Foundation, and one of the big targets of the Gates Foundation is the organism that you see there, the mosquito that carries malaria. And there had been an observation back in the mid-2000s that a drug called ivermectin that's been used for the treatment of a whole range of different infections, particularly parasitic infections, was also toxic to those mosquitoes. And they asked, could we help them develop a system that could maintain that level of ivermectin for a prolonged period, because they could see how that could help essentially reduce mosquito burden in villages as mass drug administration campaigns went through the areas where there's high endemicity of malaria. And so what you see here is the very first experiment we did with this platform, and we showed that we could maintain that ivermectin level initially out to 10 days, and by the time we published the work, we had reached 2 weeks with our system as compared to 2 days with the off-the-shelf approved ivermectin. Now, we recognize the modularity imparted by this star-like structure and actually explored a whole range of other applications. The next one was HIV, and so we developed a set or a capsule that could deliver an antiretroviral for a week we identified some potent antiretrovirals and demonstrated we could do that. And so what you're seeing here is representative data for one of those antiretrovirals where we compare the daily dose versus the week-long dose. Now, beyond that, we actually continued sort of extending the residency and release, and what you see here is a once-a-month oral contraceptive system where we show that using this system, we have the capacity to release a contraceptive over the course of 3 weeks. Now, what I want to just highlight as part of this effort, we are very committed on getting these systems to humans, and we started a company a few years ago called Lindra. And with the help of an incredible team, an incredible set of supporters, this is actually recent data from a phase 2 clinical trial. In this case, one of the main products that Lindra is developing is really targeted to those suffering from schizophrenia. And they're ensuring that the patient receives their medication can be critical. And the disease itself affects or modifies the ability of the individual to ensure that they are continuing to take their medication. So there's a few basic things I just want to highlight here. What you see in the orange and blue boxes is essentially a comparison between a daily tablet and the weekly star-based delivery system. And what you can appreciate, I hope, is that as far as the drug levels, the concentration, the minimum concentration and the average concentration using the two approaches was equal. The other aspect that I think is really important to appreciate is that the Cmax, or the highest concentration that's achieved, is actually lower or can be lower when the gastric resonant star is used. And so that starts to help us address some of the toxicity that's associated with those peak concentrations. So this journey continues, and I really hope I get to update other folks in this room and in various forms over the coming years. Let me just shift gears to another body of work actually supported by the Gates Foundation. So about 6-7 years ago, I had an opportunity to update the leadership of the foundation, including Bill Gates and some of the Gates Foundation close advisors. And one of the questions they asked me at the time, and it was actually, I was presenting the early data for the STAR system, and they asked me, they said, what do you think we should be doing? And I said, well, at the time, I had 2 young children. One was 3, the other one was 1. And I said, well, personally, I find it really challenging to help my children receive medication. And I'm a trained gastroenterologist living in Boston, so you can only imagine what it's like in some of the areas that you're trying to help. And my general sense is that there isn't that much investment or focus on helping pediatric populations. And so this sort of set up a conversation and eventually led to a major grant that really enabled the development of 2 platforms. I'm going to share one today. And this is work that was led by John Wei Lee, who's an incredibly talented postdoctoral scientist in our group. And now we're continuing through a new company called Synthesis, and Rahul Danda, who I've included there as a CEO. Now, the way that we approach this problem is a little bit different. So we wanted to develop a system that could exist in a liquid formulation, recognizing the challenge that children have in ingesting solids. And what we wanted to try and identify were cues that were unique to the upper GI tract, specifically the small intestine, recognizing that that is the main site of drug absorption and nutrient absorption. And so we identified an enzyme that is preferentially expressed in this region. That's what you see here in the form of the purple Pac-Man. That enzyme is called catalase. Catalase happens to break down hydrogen peroxide, and while doing so, liberates oxygen. Hydrogen peroxide is present in toothpaste and in certain foods. And so what we wondered was, well, could we include a tiny amount, even much lower than is present in, for example, toothpaste and foods, but sufficient to generate a local amount of oxygen that could then drive a whole reaction. The reaction that we focused in on was the reaction of the formation of an adhesive, the same adhesive that mussels, the shellfish, use to stick to rocks. And this is polydopamine, and that's what you see in that sequence 1, 2, 3, is essentially that formation of a layer, the polydopamine layer, on the mucosa. So what you're seeing in this animation here is that dopamine solution with the oxygen that's liberating, and you see that dark brown layer forming on the mucosa. Here's a zoomed in view. You see the purple Pac-Man breaking down the hydrogen peroxide, liberating that oxygen, and then you see that the dopamine then gradually forming into essentially this polydopamine layer, which is dark brown, which adheres to the mucosal surfaces. So what we were creating was a targeted adhesive in the upper GI tract. So this sort of then stimulated further exploration, but just to sort of give you an up-close and personal view of what this looks like, on the far left of the monitor, what you're seeing is a segment of the intestine. You can see the inside of the intestine, essentially is a piece of intestine that's been turned inside out, I should say, and you can see the dark brown layer, and that is essentially that polydopamine layer. We characterize the speed with which this forms, and it forms relatively quickly, and we also characterize and show that actually that deposition occurs very specifically where we expected it to, in the small intestine. And so that's what is reflected on the far right, showing you the highest levels in the duodenum and the jejunum. Now, recognizing that we had this platform, we zoomed in. What you're seeing here is a zoom-in view of the villi in the small intestine. You can see that brown deposition occurring right at the surface. And so recognizing that we had the synthetic layer, we asked the question, what could we do with this layer? And we explored three different applications. One was, could we have a biologic? Could we have an enzyme immobilized transiently and have it do its function in this area? So could we enhance, for example, digestion and potentially open up new treatments? Two, could we modulate the porosity of this synthetic lining, potentially to modulate, again, nutrition, but now absorption? So could we, in a way, create a drinkable gastric bypass, potentially to help people suffering from diabetes or obesity? And then three, what can we do for drug delivery, particularly now thinking of the pediatric population, which is where we started? So let me just walk you through what we did. So as far as the enzyme that we immobilized was the enzyme lactase, or beta-galactosidase. And what we showed was that when we included that enzyme, and this is all work that was done in pigs, that we could enhance the amount of breakdown of lactose by 20-fold as compared to if the enzyme had been administered on its own. Now, as far as the blocking of nutrients, the nutrient that we looked at was glucose. We looked at several others later as well, but we focused in on glucose, recognizing the potential benefits of such a system for patients suffering from diabetes and obesity. And so what you're seeing here is a challenge of an animal that has been administered this layer. And what you can see here is that the rise or the excursion of the glucose level is blunted in the group of animals that had been given the G cell being the gastrointestinal synthetic epithelial lining. Now, what we also know, we designed this system to adhere to the surface mucosa. What we know is that mucin and that surface mucosa turns over about once a day, or approximately every 12 to 24 hours. So if we repeat that experiment the next day in an animal, the same animals, but where we haven't dosed them, then that returns to baseline. So it's something that can be controlled from one day to the next. Now, with respect to drug delivery, which is where we had started, we looked at a drug called praziquantel, which is used for the treatment of some very specific parasites, and is a drug that is dosed 2 to 3 times a day in some big capsules, and it's very challenging for children to take. And what we showed was that if we administered the drug using the system, we could enhance the absorption, because now that drug is actually localized directly to the main site of absorption. And so what you're seeing there on the far right, as far as the exposure, the amount of drug that's absorbed, is that area under the curve, the AUC, is increased 4-fold. And effectively what this is doing is enhancing the half-life of the drug. So now a drug that used to require dosing 2 to 3 times a day can be dosed once a day. So this journey continues, and I look forward to updating everyone as this expands. Now let me shift gears to the last body of work, and this has been an incredible journey that I've had the incredible honor and pleasure of being involved in since I was a gastroenterology fellow and over there at the Mass General. There have been many people over the years that have been involved, but these are some of the key contributors over the years. And it's been an incredible collaboration, both locally, but also internationally with the Novo Nordisk team. And this body of work really focuses on the recognition that in general, most patients and healthcare providers prefer the oral route of administration for the administration of therapy. Now, what we recognize is that many therapies cannot be easily administered orally for the simple reason that the GI tract breaks those therapies down. I'm specifically referring to biologics, which include nucleic acids as well as proteins. So just before I share with you our sort of engineering efforts to help address this, I wanted to share two clinical cases. These are clinical cases that I saw when I was a gastroenterology fellow when we started on this journey. And so the first case is an extremely memorable case that came in on a Friday. And it was a gentleman in his early 40s that was transferred from a local jail in the setting of a witnessed event of shards of glass. And I was the gastroenterology fellow on call, and I was called down to see this gentleman. And as I approached him, I saw that he had a half-eaten sandwich in his hand. And I asked him to stop eating. I evaluated all his studies, his x-rays. I could see that there were actually radiopaque pieces of glass. They were about just under 2 centimeters in their maximal dimension. And I shared with him that there wasn't much that I could do as a gastroenterologist for the very simple reason that he had just eaten. And recent ingestion event essentially precludes us doing an emergent procedure for the risk, because of the risk of that food coming up and then going down into the lungs. I reviewed this case with my attending at the time, and because of the size of the sharp objects, we determined that it probably would be safe to observe this gentleman over the weekend. And this to me was mind-blowing at the time. And subsequently, I presented this in grand rounds and looked at several case series looking at the intentional or inadvertent ingestion of sharp objects. And what is reflected today in our GI guidelines actually is exactly this, that if the object is small enough, it is safe, and in general, it is acceptable to observe the patient, and that often those sharp objects will transit without complication. And so this to me was just an amazing case, and just really highlighted to me the incredible capacity of the GI tract to tolerate sharp objects. The second case is a more common case that we tend to see. This was a lady in her 60s suffering from arthritis who had been taking ibuprofen. She'd been taking it because she'd been suffering from some joint pain secondary to her arthritis. And she came into the emergency department complaining of epigastric pain, that's right here, overlying the stomach. Additionally, complaining that her stools were black and tarry, and she also mentioned that she was lightheaded. So this constellation of symptoms and signs, particularly in the setting of this history of ibuprofen intake, makes us, gastroenterologists and internists, think that there may be a gastric ulcer. And so lo and behold, we did an endoscopy, and what we saw was an ulcer that was bleeding. One of the things that we often will do in these settings is we will inject that ulcer with epinephrine, with adrenaline. And the reason that we do that is for a local effect, for a local vasoconstrictive effect, essentially to clamp those vessels down to allow us to get a good view and control of the area and then intervene with, for example, a clip or cautery. But as soon as we give that injection, all the alarms in the room start bing-bonging because that epinephrine that was injected for a local effect has systemic effects. And so putting these together just made us wonder, maybe we might be able to create capsules that give an individual an injection from within. And so what you see here was some of those very early concepts on the far left and some of the early prototypes that we actually gave pigs. And we examined the pigs, and there was no damage throughout their GI tracts, which was, again, consistent with our knowledge and the clinical experience. The other thing that we did was we obtained some insulin. So my wife is an endocrinologist and an obesity expert. She was finishing her training at the Joslin and at the Brigham at the time, and she was kind enough to help us with some insulin, and we injected different parts of the GI tract of the pig and showed that actually we could reduce their glucose and in a way that was comparable or even faster than subcutaneous administration. So this was really motivating and actually helping convey or establish a vision that potentially an orally delivered microinjector may be able to support the administration of a biologic. And so where we were, this was about 8 years ago now, 9 years ago, was there were still some major challenges in front of us. How do we get that milli-injector or microinjector to approximate or engage the mucosa in a predictable fashion every single time? Two, how do we have or how do we impart an autonomous triggering system in that device? And then how do we fit enough drug in a device that is the size of a blueberry? And we wanted to bound or constrain that size because we wanted to ensure safe transit of that device through the GI tract in a broad population. So let me walk you through how we've approached this. So for the engagement with the mucosa, we took a page from Weeble Wobbles as well as a leopard tortoise and designed a system that could self-orient, as you can see here. We evaluated the system through high-speed photography, initially on the bench, but also in the stomach of pigs under direct endoscopic visualization through simulated walking and agitation in that environment. Now, with respect to the triggering, we wanted to identify a cue that was universal to everybody's stomach. And the cue that we honed in on was humidity. And so then, what we wanted to identify was a material that could hold or constrain or store the energy in a compressed spring to facilitate the deployment of a needle. And the material that we honed in on was sugar glass. And so sugar glass, I want you to think candy cane, not taffy. It displays brittle fracture mechanics in that it will fracture all of a sudden, and therefore, as it's dissolving in that humid environment, will give way all of a sudden, providing the release, the sudden release of that stored energy. And what you're seeing here is high-speed photography, one of those early prototypes of a spring encased in sugar glass. And then finally, as far as the drug is concerned, we created solid formulations or solid needles made of up to 100%, in this case, insulin, so you can see on the right. What we demonstrated using this system was that we could achieve levels of insulin that were comparable to subcutaneous administration. And we've expanded this work more recently. We've actually redesigned the inside of the system and now can actually also deliver liquids. And so what you're seeing here is the latest version of our system where now we are still using all the same principles of self-orientation, of a material that displays brittle fracture mechanics that is sensitive to humidity, but now we are triggering the injection of a needle through which we can inject a liquid. But we can also, we have a secondary trigger that then retracts it. And so using this system, we define the optimal depth of penetration in the gastric mucosa to create that depot. And that's what you're seeing on the bottom panels is that optimization of the depth of penetration to create a discrete submucosal depot. I'm just going to skip that video, but just to highlight here that using the system, we've now demonstrated that we could deliver monoclonal antibodies as well as insulin, GLP-1 receptor agonists, and also adrenaline for that matter. More recently, we've actually used the system to deliver mRNA orally, and we've started a clinical trial just earlier this year for the first in human demonstration or application of these systems. So with that, it really takes a village to do this kind of work. This is our group at our summer party along with our families. It's really my honor and pleasure to stand here and share the work of many. In case there are any questions, I'm happy to take them, but if we don't have time, feel free to email me. I also want to acknowledge the incredible support of our institutions as well as of our incredible sponsors and collaborators. So thank you very much.